And look, I really hope we look back on this day, Rajiv, and others here at ICRISAT. I hope we look back on this day and we think of this day as the beginning of some really nice collaborations, and hopefully we can achieve some impact together. And and maybe when I'm really old, maybe we make a change in the world of plant breeding and crop productivity on farms. This would be really nice. I don't know how sometimes it's hard to predict the future. I think we're on the right track. I think developing these technologies and helping speed up the breeding programs, I think this will lead to change. So, first of all, it's great to be here and give an update on some of our speed breeding technology and how this fits in with some of the genomics tools and technologies that are being developed here. So to kick things off, I thought, well, there's actually been a lot of media around this technology lately with new papers coming out. And so I thought just to play the video from our press release rather than give the usual boring slides as introduction. So hopefully you don't fall asleep.
Well, the world population is still growing. By 2050, we'll have something like 10 billion people on this planet. So it means we've got to produce more food. But we've got to produce more food under climate change. In Australia, we're facing one of the worst droughts we've ever seen. Farmers in Europe, on the other side of the planet, are also dealing with drought and heat. So we really need to speed up the development of more productive crops in the face of climate change. Plant scientists and breeders all around the world are working on discovering new traits and genes for drought adaptation or heat tolerance that can be deployed in future varieties. The problem is, it just takes so long to put those traits into varieties for farmers to grow. So speed breeding can really help fast track this process and deliver more robust crops to farmers sooner rather than later. Normally this technology can be quite expensive to set up large-scale facilities. So we wanted to empower the plant scientists with a small budget. Our latest research paper between the University of Queensland and the John Innes Centre in the UK, we've reported the protocols on how to build your own speed breeding cabinet or DIY speed breeding.
We've also built a miniature desktop cabinet with bits and pieces that we got off the internet. It was very cheap. It only cost £700 or £800. And this tiny little cabinet now allows us to trial a new crop or a new cultivar of a crop and look at a new trait. And once we've optimized the parameters, we can then scale it up in the glasshouse.
We're hearing more and more from institutes around the world establishing speed breeding facilities to help speed up their plant research or breeding. And we're pretty excited that maybe one day speed breeding will really deliver some of our future varieties for our farmers.
I really like the funky soundtrack at the end there, But I definitely won't give up my day job, so my acting skills are not that good. So to tell a bit more about the story and a bit of a background about this technology and the development, here is a visualization of a traditional breeding pipeline. I guess this can really be representative for any inbred crop where you're crossing, inbreeding in the first phase. Then you do this very extensive yield testing and testing for grain quality, or for whether it's bread making quality or noodle making quality or whatever it is for that particular crop. Disease screening is very important for plant breeding, of course, and then you go into this. Once you've found the winner among the pack, you then have to bulk up all the seed and make it available to farmers to grow, and that can take much longer than 1 to 3 years, but depending on where you start that whole process. It can be quite frustrating to bulk up seed of a new improved variety. It takes quite a few years. But if we look at this crossing and inbreeding phase, it can be quite time-consuming, and this is really the actual breeding back end of things in terms of the breeding pipeline. If you're only growing one generation a year, it can take up to 7 years to go from cross to producing inbreds, say around the F6 generation. And this is a reality for many breeding programs around the world. It's different for each crop. There's different constraints for different species and different regions, and every breeding program is different in terms of the resources that they have available. In Australia, we have very modernised breeding companies that are actually trying to use all the tools in the shed to go very, very quickly. But you know, there's other programs around the world that don't have many resources, and so it's very slow. And this is actually a reality. However, if you do 2 generations a year, it's simple math really, you can do this whole process in half the time. And that's exactly what was achieved when Dr. Norman Borlaug introduced this strategy called shuttle breeding into CIMMYT, achieving 2 generations a year instead of 1. It literally cut the time for variety development in half. And this was a long time ago he proposed this idea. Back in the day, you can imagine he proposed to the workers that they would throw those wheats on the back of a truck and go and plant them at another location. It's kind of crazy. And he wouldn't have been very popular because it literally doubled their workload for the year. But it had a huge impact on speeding up the development of varieties, fast-tracking the whole green revolution. And this strategy was particularly successful because it enabled selection for broadly adapted material that was very successful in different parts of the world. Double haploid technology is another technology that has really been transformational for plant breeding, particularly for crops like winter wheat, maize. It's double haploid systems have been developed for pretty much all major crop species now. And it shortcuts this whole process of single seed descent, going through the generations of selfing to get homozygous or inbred lines. You make the F1, you double the chromosomes in the lab using colchicine or other chemicals, depending on the methodology. And so you can generate stable lines very, very quickly. And some of the techniques that have been developed and highly refined in specialized labs around the world, and they're even faster than 2 years. They're down to about 1 year or less in some cases. And so this is actually, I visited a lab in Germany where they have really refined double haploid systems. Recently, earlier this year, they service many of the major breeding companies there in Germany. And even though this is a fantastic tool, there are limitations to any tool, right? And so one of the limitations for this is cost. And for wheat, for instance, it's still costing about 12 euros per line, per double haploid line, and half of that for barley. So it's about 6 euros for a barley double haploid line. And that's pretty expensive when you're talking about generating thousands and thousands of lines in a breeding program. I remember I had breakfast with Hans Brown once, and he asked me, well, he was trying to weigh up whether to go down this single seed descent rapid cycling path in double haploids. And he asked me in his very strong German accent, I can't do it very well, so I won't attempt, what's the cost of generating 100,000 double haploid lines? And it's pretty costly. You can develop this skills in-house, but you have to have specialized labs and facilities to do this, or you have to outsource it. Other downsides is that you don't get as much recombination. You only get recombination equivalent to the F2 generation, whereas if you do the single seed descent process, you get all that extra recombination. And let's face it, recombination is a very good friend of a plant breeder because it's all about coming up with all those improved or better gene combinations. And of course, you don't get the opportunity to apply any early generation selection. The process is random. You get what you get. And the efficiency varies across crosses. And so there are some limitations. It brings me to perhaps an alternative, although I do argue that these technologies can be fused. More on that later. But first up, I wanted to just mention about where we actually got the motivation or inspiration from around this whole rapid cycling or speed breeding idea. And it was originally from NASA-funded research that was aiming to grow wheat in outer space. And this was an effort funded through the Utah State University back in the 80s. So it was quite a while ago. And the way that they were doing it were they were growing the wheat very fast by exposing the plants to continuous light the extra light triggers the reproductive phase, and they produce seeds very, very quickly, much faster than normal. And so we thought, what a cool tool this could be to help speed up our crop research and breeding, perhaps, here on Earth. And so, interestingly, they actually developed this variety called apogee, first food product developed specifically for growing in space. And here it is. These are images released this year in March or so by the International Space Station of wheat growing there on the space station for the very first time. And this is the size of a small beer fridge. So it's very small space. There's not much space in space. And so they literally had to develop this variety as a full dwarf. You can see how short it is. It's about literally this big, and it's mostly spike. So it's purposely designed. So we thought, this is a pretty cool idea. So we took this idea and we started trialing this speed breeding or rapid cycling system at the University of Queensland. This is about 15 years ago with my colleagues. And look, when we first tried it, it was the first time we started growing wheat in the glasshouse, and we failed miserably. The plants were terrible. We had no idea. It's the first time when you grow a plant, you need to get the nutrients correct. But once we optimized the temperature regimes and the photoperiod and the nutrient supply and the media, the plants grew great and even resembled the plants in the field. So we now use it routinely for all of our pre-breeding and research and breeding with industry. And you're looking at here at our first generation speed breeding system. For more than 10 years, it looked like this, and it worked very well. Over the last sort of 12 months to 18 months, there's been a massive spike in interest in rapid cycling or speed breeding, and I have to say it's probably because we're all under pressure to do things faster these days. Time is of the essence, no matter what you're doing. Whether it's in the research world, to publish papers faster, to do the training faster, to speed up our breeding efforts. Everything's all about time. Time is money. And so we've just got inundated completely by requests from all around the world. We got more than 200 requests from scientists and breeders from around the world for our protocols. And so I started replying to each of them one by one via email. And I'm writing these 4 or 5 page emails, and it was so time consuming. I mean, I wanted to share the information and tell people how to do this, but it wasn't probably the smartest way in terms of time for myself. And so we decided to team up with other groups who incidentally have developed their own speed breeding systems. And so we teamed up with the John Innes Centre and the University of Sydney to actually publish and make these protocols available. And so earlier this year in January, it featured on the cover of Nature Plants, which was just fantastic exposure, and really put the spotlight on this tool for crop improvement. And before I go any further, I really need to thank and acknowledge the key people behind this amazing duo, Amy Watson, my PhD student, and Shreya Ghosh, PhD student of Brando Wolf. These two have been the real powerhouse behind these 2 publications, making all this information available to the world. And they're the 2 really bringing all these massive data sets together. We've accumulated so many data sets from so many people. Brando Wolf has been the brains behind setting up speed breeding at the John Innes Centre, and really driving all this with myself. And of course, Robert Park's group at the University of Sydney. So, in a nutshell, this is what we can do with speed breeding. We're actually refined the methodology, so we're giving the plants 22 hours of light, not 24 hours. So we actually used 24 hours for more than 10 years. But we found that through some of the experiments actually conducted by the John Innes Centre, the 2 hours dark phase actually improves the quality of the plants. That's the great thing with teaming up with centres like the John Innes Centre. They literally, for speed breeding, they literally science the shit out of it. They're really good at doing the science. We were applying the technology, but they took this to a whole new level in terms of doing the science around proving how effective this tool was. And so we could generate 6 generations, do 6 generations of wheat per year. This is spring wheat. 22 hours of light per day. We actually nice to them. We give them 2 hours of dark. And for that 2-hour dark phase, we cycle down the temperature to 17 degrees. And so Amy Watson, my PhD student, ran a whole bunch of experiments as well at UQ, and she compared the growth of a whole bunch of species. And it works out that it works, this system across wheat, barley, chickpea, canola, and we can get 6 generations of barley and chickpea, only 4 for canola, because it takes a bit longer to harvest the physiologically or viable grain or seed from the canola plant, because they're quite oily. It takes a bit longer to form. And this compares to just 2 or 3 generations in a regular glasshouse. 3 is at best. Sometimes it's 2 per year, because it just depends on the seasonal conditions you're subjected to in a regular glasshouse. Just to highlight how fast these plants grow, our collaborators captured this beautiful time lapse, because seeing is believing, right? And so on the left, we have speed breeding, wheat plants. On the right, normal breeding, regular glasshouse conditions. And you can see just how fast they grow. The wheat under speed breeding, it's already elongating. Now it's flowering, filling that grain, and approaching maturity and harvest time. While the normal breeding plants are just far slower, and we don't have time to finish the cycle. It's just too slow. And so just to capture, these images are beautiful because they capture the growth rate differences we see in speed breeding plants versus regular glasshouse. And these plants were sown on the same day in glasshouses side by side, but one was fitted with a supplemental lighting for speed breeding for 22 hours and the other not. And these photos were taken after 3 to 4 weeks. And so all the speed breeding plants are essentially flowering compared to these regular glasshouse plants, much, much slower in development. And it turns out that many of the speed breeding plants actually produce more seeds and were more productive under the speed breeding conditions, which is surprising to many people too. I guess it's because of the extra light that they're getting. But it works for a whole bunch of species, and almost weekly, this list is growing. So it works for pea, even the hipster food, quinoa. A lot of hipsters in Australia like this. Grass pea, orphan crops, there's a big potential there. Peanuts, or ground nuts. It works for peanuts. So the peanut breeding company in Australia now runs their whole breeding program through speed breeding. And they achieve 3 generations a year instead of 2. And in their words, they say, or Graham Wright, the breeder there, says it's probably the most impactful thing that they could have done in their breeding program. But it also has an impact on the actual crop research too, with many model species being accelerated through this speed breeding approach. So it can help speed up the research. The list is continuing. According to the Twitter sphere, Carl Schmidt, a very good scientist in Germany, brought to my attention that they actually developed a very similar speed breeding protocol for amaranth. And this is really interesting because amaranth is a short-day species. And, well, how did they get it to work? Well, they developed this nice recipe, if you like, where they're exposing the plants to long days in the vegetative phase, then apply just a few days of a short-day photoperiod to trigger flowering, and then finish off the cycle on long days. And so by using this approach, they can achieve up to 8 generations a year for amaranth. So I dare say that speed breeding is possible for any species. We just got to work out the right parameters, and the protocols need to be optimized to achieve that. Here's lentils. This is a collaboration with the University of Melbourne, and here's a cultivated lentil our farmers currently grow. This is a landrace that normally takes 180 to 190 days to flower under regular conditions. Under speed breeding, 27 days to flower. So this tool can open up whole new gene pools for crossing and bringing in new traits, new diversity into our modern crops. The speed breeding recipe is truly flexible. And I think that was really highlighted in our paper, because when we brought together these protocols from the University of Sydney, from the John Innes Centre and UQ, we had different systems. You know, at UQ we had just temperature-controlled glasshouses with, at that stage, our old generation lighting, the sodium vapor lamps. At John Innes, they originally set up speed breeding in these very expensive Conviron growth chambers, and they have a lot of money, the John Innes. They've got a big bank of these things. And the University of Sydney, they had actually, it was pretty cool. This is a pretty low-cost, innovative solution where they just had a regular office room with an office air conditioner with some LEDs. And they were using this to accelerate their back-crossing efforts for transferring rust resistance much faster. One of the innovations, or more recent innovations around reducing the cycle time even faster, has been around early harvest of the seeds. And so we can actually harvest in wheat and barley. We can harvest these seeds just 2 weeks after anthesis. We dry them down in a dehydrating oven for a few days, and they look like this. They're shriveled. Yeah, they're not fully formed, and they don't want to germinate, however. But we have a trick for that. Alternatively, we can wait an extra 2 weeks and reduce the watering, and they mature naturally, or more naturally. And so the grains are more fully formed. And that's great if you're going to look at any sort of grain traits or grain dormancy, as I'll mention later. To get those early harvested seeds to germinate, and I think this is really cool. We worked out that if you apply a cold treatment to the seeds, they all germinate. So we imbibe them for 24 hours overnight at room temperature. They soak up the water. We pop them in the fridge at 4 degrees Celsius for 3 or 4 days, bring them out, and we get 95% germination. And all this can be scaled up to literally hundreds of thousands of seeds in commercial or big breeding programs. And what this means is we're actually harvesting the grain at the same stage as embryo rescue methodologies. But we don't need to go through the lab. The lab sucks. So if you're a plant breeder, the lab really sucks because it's a real bottleneck. So every time we have to go through the lab, this is a bottleneck, and I think this has been one of the key innovations to reduce the labor around applying these sorts of rapid generation systems. So if we look back at our really boring breeding pipeline figure, this is the crossing and inbreeding phase, and speed breeding is all about shrinking that down. Doing the crossing and inbreeding much faster means we can test these materials faster in the field. And so there's a huge potential to increase genetic gain with these sorts of rapid cycling systems. So Dr. Norman Borlaug's favorite equation, the genetic gain over time equation, and you're all probably very familiar with this. We want to increase all these upper components of the equation, like selection intensity, selection accuracy, and genetic variance to some extent. There's trade-offs, but speed breeding, like shuttle breeding, fits into the denominator of the equation, which is the most powerful part of the equation, and I would argue has been somewhat ignored until now. And of course, genomic selection also targets this component of the equation. Apparently, there's people are starting to call me or give me a nickname as the denominator. I don't know whether I like that or not. It's exciting because we've been working now with the speed breeding technology with industry for quite a few years, and the first wheat variety has been released in Australia that's been purely bred and selected under speed breeding. This is called DS Faraday. It represents Australia's first high-protein milling wheat with tolerance to pre-harvest sprouting. And this was released in partnership with Dow AgroSciences. So this is the best wheat for making bread. And we managed to introduce this completely novel trait in the market. All of our wheats are white-grained in Australia and completely susceptible to pre-harvest sprouting, so rain at harvest time. And so how do we do that? Well, we didn't have markers for all the many genes that are needed to give this 2 or 3 week grain dormancy phenotype at physiological maturity. So we selected based on phenotype. We transferred what we suspect to be between 10 and 12 genes controlling grain dormancy from landrace material into this elite cultivar with premium grain quality. And we did this in about 20 generations in the speed breeding system, coupled with phenotypic selection for grain dormancy. So we actually had to grow the plants to physiological maturity. This slows us down. We could only do about 4 generations in the speed breeding system because we had to get this true phenotype. If you sample too early, you have this imbalance of a bias phenotype of ABA imbalance in the grain. And so we managed to actually transfer this. So this is pretty exciting. I think it shows the potential of what speed breeding can do if we think about other traits important for climate change and disease resistance. And so what other traits can we measure under speed breeding? Well, we showed nicely in our publication with the John Innes Center that we could phenotype a whole bunch of traits, simple traits, often underpinned by, single genes. So all morphology, of course. There's some of the green revolution genes, flowering time, alleles, waxiness. All of these are beautifully expressed. And so you can see how this might be able to really speed up some of the research or pre-breeding, particularly if scientists are working on adult traits, where you need adult plants to phenotype some of these characteristics. Spike architecture, those sorts of traits. One of the physiological traits that my team are really working a lot on, because drought is a big problem in Australia, water is very limiting, is root architecture. And so in our part of the world, we have deep soils, terminal droughts are quite frequent. And so if we can design wheat varieties with deeper root systems, they could access more stored soil moisture deep down in the soil profile. And so one of the proxies we're looking at is this trait called a narrow seminal root angle. And Cecile Richard, one of my ex-PhD students, she developed this really nice system called the clear pot method. It's not rocket science. It's literally a transparent garden pot. And we sow the seeds around the outside, and we can visualize this root growth angle. And we measure the root angle using an iPhone and some open access software. And we can actually apply selection. We use this technique to track down some of the genes for mapping populations. But Cecile applied this technique in combination with speed breeding to actually apply the first direct phenotypic selection for root architecture in wheat. And so she applied bidirectional selection. So she selected for the plants with the widest root angle and the narrowest root angle, and effectively shifted, after a few cycles of selection, shifted these mean distributions. So we generated these BC1 F4 derived lines in several different genetic backgrounds, elite backgrounds, and we did all this in just 18 months. So not only did we show that we could manipulate the roots through direct phenotypic selection, and once we get the markers, this will be great too, but we generated this highly valuable pre-breeding germplasm that we can go now to the paddock, to the field, and actually try to validate some of the value of actually having these modified root systems. And that's really important to demonstrate the value of these traits if they're going to be picked up by plant breeders. So roots are a pretty hot topic at the moment because they're underground, right? And we haven't really harnessed them in terms of plant breeding very well, not as much as the above ground traits. And so we actually have a couple of new projects. We have a new IWIP project with NIAB in the UK with lots of different partners, and a new project in Australia. And with these projects, I mean, we're taking different approaches. We're using CRISPR to target some of the key genes, but we're also just taking the traditional breeding approach and rapidly backcrossing these traits into elite varieties using the speed breeding technology. So this is helping to fast track a lot of our root research, and hopefully we can have an impact with these traits because, like I said, not much is being done in this department. A lot of my team is also focused on diseases that are a big problem, and so they've developed really nice protocols for actually screening for disease resistance in the speed breeding system. So that way we could do rapid screening for disease all year round, and so this is really helping to speed up our research and pre-breeding. So for instance, one of my ex-PhD students, Eric, developed a nice system for yellow spot, actually the number one foliar disease in Australia for wheat. So this is all these systems we're trying to develop in the speed breeding system are around adult plant resistance because there's greater value in speeding up the rate of development to look at these adult traits. And Adnan, he's the rust man. He developed some really nice systems for leaf rust, stem rust, and stripe rust as well. And so here, this was work done at the John Innes Centre, showing nicely that we could screen for Fusarium heblite resistance in the speed breeding grown plants. So this helps speed up some of the work that we're trying to combine the genomics to track down some of the genes controlling these key traits. One of the collections we're working with is the Vavilov collection we assembled. And so as a pre-breeder, we're really interested in bringing in new diversity for traits like disease and drought. And so this Vavilov collection, a lot of my lab is working on, and we're using the speed breeding system to rapidly track down some of the key genomic regions responsible for these traits through GWAS studies. But we're also then generating secondary validation populations for fine mapping. But in parallel with this, we're also starting the rapid backcrossing phase. So the idea is we bring all these things together rather than do them stepwise. So now, for instance, we've just finished developing a whole suite of introgression lines for yellow spot and rust in all the elite varieties that farmers currently grow. And we did all this work in just 3 or 4 years. So this is really helping us to accelerate things. When you can do 6 generations a year, I mean, it really helps to fast track everything, including student training. The students get the whole experience of generating their own populations, making the crosses, generating a real population, doing the genotyping, doing the phenotyping, and that just wasn't possible before. Students often got handed, sort of someone else's population to work on because they didn't have time to develop it. Speed breeding 2.0. Maybe some of you have heard Ed Buckler speak. He's got breeding 1.0, 2.0, 3.0, 4.0 is where everything goes crazy. Well, speed breeding 2.0 is kind of like that, but this is, I think, centered around reducing the operating costs and scaling up this technology. And so one of the ways we're doing that is we've just replaced all of our old lighting with the latest generation LEDs, and so they're way more efficient at reducing the heat load in the glasshouse. So that's reducing energy to actually fuel the lighting, and it's also reducing the energy required to cool the system. So it's way more efficient. The lights are only producing the wavelengths that the plants need for healthy plant growth. And so we're actually using these heliospectral lights. We find work really well. They plug and play really well. They provide a good service, but there's lots of other companies that also provide very similar lighting, which I'm sure work just as well. What you're looking at is a drone bird's eye view of the speed breeding facility now scaled up at the John Innes Centre. And it's interesting because when Brando, my partner in crime, in speed breeding crime, suggested that they do this speed breeding stuff at the John Innes Centre, they were very skeptical, and no, they're scientists. That's fair enough. But once they did all the experiments and they showed how fast the plants grow and how healthy they were fully convinced, and everyone wanted to do speed breeding. So now they had to set up several of these massive glass houses for all of the researchers to run all their research material through. And so luckily, we've just managed to publish the next paper in Nature Protocols, just released about a month ago or so, which is a detailed, we call, complete A to Z guide of speed breeding. Well, I say that it's not a complete guide because we don't have all the species in there, but the way it's written is that it's a bit of a stepped guide on how to troubleshoot and tailor these systems to your own species of choice. So hopefully it's very useful to adapt speed breeding to your own crop. And of course, we included a whole bunch of more crops in this, but it was just a huge effort. There's so much information in this paper, it's ridiculous. There's tables looking up tables. There's that many tables. So yeah, check it out if you're interested. And I couldn't help but edit the cover of Nature Protocols because it's the festive season, so I thought it needed a Santa Claus. But yeah, like I said, there's all the details in there for setting up speed breeding, at least on how we've set it up at UQ and the John Innes Centre. I'm sure there's other ways you can adapt these systems to other environments. I think it's even possible to I think it might even make sense to do this sort of speed breeding even in a closed environment, like in warehouses, like in environments here in India where it's very hot, to reduce, because in glasshouses you get all that extra sun, it's very warm temperatures, and I think that's exactly what Pooja is working towards, and Jan are working towards setting up here at ICRISAT. So that's pretty exciting, and hopefully it'll accelerate a lot of this genomics work that's going on and breeding efforts. In terms of scaling up, like I mentioned a warehouse, and it's exactly what you want to do, right? This is about mass production of plants, and it's a factory. It's a plant factory. You want to produce plants as cheap as possible, a few cents per plant if you can. And so it's about packing in as many plants as possible. And so we ran some nice experiments in these Nature Protocols papers just to show how you could do that. For wheat and barley, here's barley growing at different densities. This is 1,000 plants per square meter, and this is 300 plants per square meter. And you can see what it does. It increases the competition when you grow them at high density. There's a reduction in plant height. There's more nutrient stress, but that's fine. We don't care. You've got to be a bit mean to them. And in fact, when you grow them at this density, it actually pushes them through a little bit faster as well. When we look at the seeds and the spikes, they're also different. The plants going at high density produce smaller spikes on average, but they're still producing 6 to 8 or 10 seeds per spike. That's more than what we need. And so I know commercial breeding programs who are actually taking this number and multiplying it by 3. So they're actually growing 3,000 plants per square meter. And when you compare that to something like the field, we don't grow anywhere near the plant populations like that in the field. So I think, the future of speed breeding could in fact be warehouses where you're growing these sort of plant factories. And if you think about it, this sort of system is perfect for combining with genomics and predictive-based breeding. One of the other aspects which I think was really cool in our Nature Protocols paper just released was this innovative DIY speed breeding effort from these early career researchers in the UK. They worked out how to make your own speed breeding system for just £800, buying bits and pieces off the internet. Like, that's crazy. But they worked out how to do it. You got everything's published, so you got the code and how to program all the lighting. And so this is critical for enabling access of this technology to everybody. No matter if you're a plant scientist with a super small budget, this will help fast-track development of near-isogenic lines, transgenic materials in labs, and hopefully accelerate crop research across the board. So I think more and more needs to be done and technology is fast evolving. I think it'll be even cheaper to do these sorts of things in the future. Can we combine speed breeding with other technologies? Well, this is where I get really excited because this is what I'm really interested in, and I see huge potential. In our paper with the John Innes and the University of Sydney, we showed really nicely that transgenic pipelines can be accelerated, incorporating the speed breeding. So we need to stop thinking about these technologies as separate and start fusing them together. That's where the innovation lies. That's where the big gains can come from. And so here you have thallus produced from embryos, grown in the speed breeding system, works fine, transformed, and here they are grown under speed breeding and regular glasshouse. And what does it do on a standard transgenic pipeline? Well, it's saving about 3 months. So, in fact, the University of Sydney are also combining speed breeding with double haploid systems. So, like I said, it's not double haploids or speed breeding. Why not both? And so what does this mean? Well, hopefully it means new traits into farmer fields faster. And, of course, some of the transgenic technologies are not accepted, unfortunately, in some parts of the world. But hopefully that could change through some of the policy and regulation around CRISPR, the applications of CRISPR. And, of course, for the academics publishing that Nature paper, very important. Genomic selection. There's some really nice work being done here at ICRISAT to develop genomic selection in our crops. And just a visual representation of what that technology can achieve. Here you have a crop breeding cycle, crossing, inbreeding, testing in the field. you're selecting the best, and off you go to cross again in the next cycle, and you're releasing varieties. Genomic selection shortcuts that whole field testing component. Of course, you still need to do field testing. That doesn't change. You need to do this for variety release, but also when you're generating the data each year in the field, you're filtering that back into your prediction models and maintaining those. So genomic selection has emerged as a really powerful tool. In fact, commercial breeding companies have been applying genomic selection now for more than 15 years, and they're making billions of dollars. So it works. We've got to get the right systems in place to be able to implement genomic selection in some of our crops. If we add speed breeding to the whole recipe here, what does it do? Well, we can crank this whole thing much faster, and that's pretty exciting. Just to sort of get a bit of a taste on what this sort of impact could lead to through combining speed breeding and genomic selection, we did some very preliminary simulations, very simple in terms of making a lot of assumptions. What we did is we took the current standard CIMMYT breeding program or strategy, and we just threw in genomic selection and threw in speed breeding. And Kai Vosvals, my colleague, simulated those scenarios. This was work together with the guys at CIMMYT and also at Huddersleibig University. And what you can see after 30 years in the simulations, you end up with this massive difference in genetic gain by combining speed GS, we call it. You've got to have a cool name these days, you know. Speed breeding with genomic selection versus conventional breeding. And, you know, you don't see the differences early on. In fact, if you look at after the first cycle, this is insignificantly different. But you end up on a completely different trajectory. And after 30 years, it's a long-term gain these technologies can help drive. And what does it mean? Well, Kai, just for fun, he did the sums and worked out what does that mean in terms of varieties that are coming out that pipeline, the SpeedGS pipeline? Well, it means varieties that are capable of producing an extra 0.5 ton per hectare. And that's 1,000 loaves extra of bread per hectare. So we talk about feeding the world. Well, this is a great example, I think, of how we can actually combine these technologies and develop more productive cultivars. But saying that this is very, very basic simulations we've done so far. I think there's even bigger gains for even more dramatic improvements through redesign of breeding programs. And we're looking at more deeper simulations to look at these things. Speaking of Kai and Ben Hayes, that is, he's actually the co-inventor of genomic selection, and I work with him at UQ quite a lot. We have an exciting new project working with industry where we're combining speed breeding with artificial intelligence. This is pretty exciting stuff, and learning a lot working with Ben. And we're asking this question, well, we're trying to address this question, how to stack disease resistance, drought, high quality, into new high-yielding varieties in the shortest time frame possible. This is a generic question for plant breeding. This is not specific to wheat. And I guess to try a different approach, we're using this fast stack approach, we call it, where it's combining the AI, guiding the genomic selection process, and coupled with the speed breeding. What does this mean? Well, this means that we can theoretically do 3 or 4 breeding cycles in just 1 year. So how long was it to do a breeding cycle before? 6 years, maybe? We're talking about 3 months, 4 months for a breeding cycle. That's a complete game changer for plant breeding. And in fact, if that actually was possible, and we demonstrate that what sort of genetic gain could come from those situations, it could be transformational in terms of plant breeding, but it would completely redesign or change the way a breeding program looks. So I think we've got to think out of the box in terms of, you know, how can we breed better? So we actually published a paper recently in TAG. This is with Wayne Powell. He has a real interest in orphan crop breeding, and look, we threw this idea out there. How do we deliver speed breeding to some of the remote regions of the world or resource poor regions of the world? And look, we thought you could actually modify second-hand shipping containers with solar panels on the roof, fit it out with LEDs, and you could rapid cycle. Now, it may not look like that in the future. It could be completely different. I don't know. But I wanted to just create some ideas or get other people to think of ideas about how we can adopt these sorts of technologies, even in developing countries or for crops that don't have many resources. Opportunities and challenges, well, there's plenty of those with these sorts of new technologies. And I have to say that commercial breeding companies are already using speed breeding. In fact, one of the breeding companies or leading breeding companies in Australia have been doing it for many years now and have what they claim to be one of the world's largest speed breeding facilities. But if you're not speed breeding, you need to invest. You need to invest in infrastructure. But I really believe that this sort of tool is so powerful that it's going to pay off, after just a couple of years, and it's going to be long-lasting. Can we take speed breeding off the grid? So with this technology, electricity and power is the key to reducing the cost of operation. And I think, with the LEDs are helping achieve this, but there's all sorts of technologies coming online. There's even solar panels that let some wavelengths through for plant growth, but capture other wavelengths for energy production that then can be used to power the chilling environment. So there's all sorts of technologies, battery storage, and lighting technologies. There's even laser lights now. So we thought LEDs were cool. There's laser lights that you can position outside the growing environment, and they beam in, and they hit a diffuser inside the growing environment and go across the plant. So that means you can take the whole heat load outside the environment. So this is rapidly evolving tech, and I don't think power will be a limitation in the short to medium term. Like I said, maybe it's better to do this speed breeding business in warehouses, indoors, in these hot environments. And I think that's a great idea to reduce the costs. Winter crops, well, they're problematic still. They require vernalization. So winter wheat, winter barley, winter rapeseed, they require a vernalization treatment of 6 to 8 weeks before you can speed breed them. So it sort of slows you down. It's a bit of annoying. I know many groups around the world are trying to address that biological issue and actually overcome this vernalization problem. So there's speed vernalization, they're calling it. But that's good. It speeds up the process. But what we need is a solution to completely overcome this vernalization problem. And we can rapid cycle these winter crops just as fast as spring crops. And I believe that there's so many people working on this that there will be a solution within the next year or two. BGS, well, you saw a bit of a taste of what it could do, but I really think that there's massive gains to be made through program redesign, rethinking what a breeding program looks like. It doesn't replace field testing. Field testing is critical, and that's actually the most important thing. But it's the back end, the engine room of combining the better genetics, using the genomics. That's where the redesign comes in. There's a good opportunity to combine this all with genome editing as well. And I really hope that some applications of genome editing are approved as non-GM. And I think it's headed that way in Australia in terms of inducing deletions, which would be the same or similar as mutation breeding. And so if that's possible, certainly there's a good opportunity to reverse alleles in the crossing cycle. But that's obvious. What if we were able to take genome editing out of the lab? What if we could do it directly in the speed breeding process? I think that would change the game. What if we could edit tens of thousands of plants at once? So we've got a couple of PhD students working on some pretty out there ideas on how we can actually implement this genome editing on a big scale where we can effectively do 3 breeding cycles a year in the speed breeding with predictive-based breeding and gene editing all at once. That's the dream, but there's a lot of research and science that needs to be done to get us at that point. So I'm kind of the face of speed breeding, but I have to say that I certainly don't do all the work, and I only do a fraction of the work, and all these amazing people do all the work. And so I'm just thankful that I can present all of this on all of these institutes and people involved in all the publication of all the speed breeding protocols. And I have to really acknowledge the funding from my side, the Australian government, and GIDC. This is money from the farmers. So there's real pressure for us to, as scientists, to have impact on the farmers' field. I feel that pressure, and hopefully we can deliver. And finally, a funny picture of my crew in Australia. Thanks very much.
Fantastic. I think super. So I know that many people may have a lot of questions, but I'm not sure whether we can take those questions right now, Hickey, Lee. So we can have those things at a later stage. So let's